Hello everyone, I'm Brandon Graham, and my proposal is titled The Impact of Recessionary Economic Uncertainty on Salivary Cortisol Concentrations. Now for the introduction. The question, does brief exposure to recessionary economic content produce a measurable cortisol response? Prior work links both lab stressors and real-world financial strain to elevated cortisol, but no one has isolated recessionary media in a controlled experiment. This study would close that gap. Two variables. The independent variable is the type of economic media, recessionary, versus the content-matched neutral control group. The dependent variable is the acute stress response, measured as pre to post change in salivary cortisol in nanomoles per liter by ELISA. Three lines of evidence support this prediction. First, psychological stressors raise cortisol. Ingrit and colleagues 2017 replicated this with their Trier task, and a 2022 meta-analysis by Go pooled 79 such studies and found cortisol 2.5 times baseline. Second, real-world economic strain raises cortisol too. Coates and Herbert saw a 68% rise in London stock traders, and Hamilton and Steptoe 2025 linked financial strain to neuroendocrine patterning. Third, recessionary content signals uncertainty and loss. Both established stressors per Peters, Garrett, and Ho. So the prediction follows. Recessionary content should engage the same HPA axis response. Hypothesis: recessionary content should activate the HPA response and increase salivary cortisol concentrations relative to a neutral control group. Between group study design with a random assignment to one of two conditions. The experimental group sees recessionary package, and the control group sees a matched neutral package. Same modality, same duration, only the framing changes. The scientific hypothesis: under controlled laboratory conditions, healthy adults exposed to recessionary content should show the same HPA axis response seen in lab and field studies, a measurable acute cortisol increase relative to the control group. Methods: 120 adults, 60 per group, recruited by convenience sampling through email list reviews, flyers, and community boards. Ages 18 to 55, balanced for sex, ethnicity, tracking the US population. Sample size came from a G-power analysis, medium effect, alpha 0.05, 80% power, which gives approximately 42 participants per group. I rounded up to 60 for attrition, exclusions for endocrine disorders, and any medications that could confound the cortisol results. Cortisol is collected with a salivette kit and analyzed by ELISA at a certified laboratory. Each stimulus package has 3 matched parts: a 10-minute audiovisual compilation, 15 infographics, and a 500-word vignette. Both packages are certified by independent economists for validity. Participants also complete the perceived stress scale. Sessions run noon to 4 to avoid the morning cortisol peak. Participants arrive, they sign consents, screen in, complete the perceived stress scale, and give baseline saliva samples, T-1. Random assignment follows. They view the stimulus package in a private room with headphones, then wait 20 minutes, the window when cortisol peaks, and then we take collection T2, and the primary outcome is T2 minus T1 in nanomoles per liter. All procedures will go through Liberty's IRB and follow the APA code. Every participant is debriefed with counseling referrals available if needed. And if it works, this is the first experimental causal test of recessionary media on acute cortisol and could help further research in the area. And these are references for the proposal. And references continued. Thank you all, and I look forward to our discussion. Thank you.